Hi guys, my name's Jordan, and me and my lab partners Douglas, Shelly, and Bo are here to tell you all about bugs as drugs. Now, let me start off by saying, when you think of bugs, you probably are thinking grasshoppers, beetles, spiders, worms. No, we're not talking about that. We're not, nope. Okay, what we're talking about is microbes. The title should really be Microbes as Medicine, but, it's whatever. So, if you're wondering what microbes are, they are microorganisms that are too small to be seen with the naked eye. Microbes are literally everywhere in our world. They live in the ground, in plants, they thrive in ponds and streams, and inhabit our oceans. Microbes have even adapted to live in extreme temperatures. There are 5 different types of microbes: bacteria, viruses, fungi, and algae and protozoa. Crazy. Most people associate bacteria and viruses with illness that commonly makes us sick. However, microbes typically do more good than harm. The benefits of microbes to our world are vast. So, examples of microbes in everyday life. The yeast used in bread, beer, and wine, yes, this, is a fungi. Bacteria exists in our intestines even to aid in digestion. Yes, it's in your body. They are in your body. Bacteria changes milk to cheese and is also found in yogurt. Now that you know the common uses of microbes, did you know that microbes have been used in medicines for decades? Some of the most groundbreaking advancements in medicines are due to metabolic engineering of microorganisms. Crazy!
Origins of bugs as drugs. Humans have used microbes to improve their lives for centuries. Antibiotics were likely the first use of bugs as drugs. Traces of tetracycline have been found in human skeletal remains from ancient Sudanese Nubia dating back to 350 to 550 CE. Traces of tetracycline were also discovered in femoral midshafts of the late Roman period skeletons in Dhekelia Oasis, Egypt. The intake of this antibiotic in these populations likely had a protective effect because the rate of infectious diseases documented in Sudanese Nubia populations was low, and no traces of bone infection were detected in the Egyptian samples. Herbal therapies that included microbes have been found in medical texts dating back more than 1,000 years. For example, bald's eye salve was found in Old English medical texts from the medieval times. The recipe included onion, garlic, bile from a cow's stomach, a bronze vessel, and wine. The ingredients were mixed together, then left to ferment over 9 days. After the 9 days, it was used to treat eye and staph-type infections. The use of antimicrobials to produce cancer chemotherapy is one of the most successful forms of chemotherapy in modern times. The creation of chemotherapy started in the 1900s. A German chemist by the name of Paul Ehrlich made it his mission to develop drugs to treat diseases. He coined the term chemotherapy and defined it as the use of chemicals to treat disease. At that time, Ehrlich's research led to the development of arsenicals to treat syphilis. He was also interested in drugs to treat cancer, but it was not until the 1960s that chemotherapies were starting to be used as cancer treatment. Chemotherapy uses chemical substances that act electively on cells in mitosis, and antimitotic agents aim to destroy cancer cells. The commonly known bacteria, E. coli, naturally expresses several enzymes that have been utilized as prodrug activators. The enzymes convert non-toxic prodrugs into toxic drugs. Insulin was first produced in the 1920s by scientists Frederick Banting and Charles Best. The scientists discovered that insulin was a hormone that diabetics lacked, and they figured out a way to harvest insulin from the pancreases of pigs and cows. After the injections of the animal insulin, diabetic patients noticed improvement of their hypoglycemia within minutes. The discovery is one of the greatest medical achievements to date. A problem still remained. Some of the patients were allergic to the animal insulin. While it is similar, animal insulin is not the same as human insulin. It was not until the 1980s that scientists discovered new technology to refine the process of producing genuine insulin by using the power of microorganisms. One of the first biotechnological breakthroughs was the production of insulin. Inside bacteria are tiny rings of genes called plasmids that determine which substances the bacteria will produce. By splicing in the chemical sequence of human insulin and then inserting this modified plasmid into an E. coli bacteria, scientists created a tiny insulin factory. It multiplied when fed, creating more of these factories until a river of insulin could be produced. Similar to beer, insulin is now brewed in large vats called bioreactors. With the use of microorganisms, it is much cheaper to produce insulin than to extract it from animals. There is also no threat of shortage or of patients having a bad reaction.
The modern era of bugs as drugs. The process of engineering microbes for the use of medicine has evolved from the early uses of herbal therapies to sophisticated scientific technology. And below is a little diagram to kind of show how it has evolved over the time. The microbe-derived medicine paclitaxel is used to fight off cancers such as ovarian, breast, and lung cancer. Paclitaxel is derived from Nodosporium sylviforme. This is accomplished by the microbe inhibiting the microtubules in mitosis and preventing the chromosomes from splitting. Eventually, this will lead to the apoptosis of the cell and cancer is eliminated. Microbes are used in other ways too. Microbes are used to lower LDL cholesterol. It does this by the inhibition of the enzyme HMG-CoA reductase. Anti-cancer, made by Trametes versicolor. It produces a polysaccharide K. Immunosuppressive, Azari sinclari, used to keep lymphocytes and lymph nodes and keeps it from interacting with the CNS, used typically to cure multiple sclerosis cases. Malaria prevention, Metarhizium anisopyle. This kills the disease inside the mosquito before it gets to the human. Inhibiting immunodeficiency. Boletus edulis has a large impact on HIV-1 reverse transcriptase. And then warding off neural diseases. Hericium coralloides suppressor of the effects of degenerative diseases such as Alzheimer's. And there's a list of different types of genetically engineered microorganism medicines. From microbes, we can get antibiotics, anti-cancers, antivirals, antimicrobials, antioxidant, antiparasitic, anti-tumor, blood-related agents, cardiovascular disease medication, hormone replacement, immunological agents. And bugs have been used in chemotherapy or in replacement of chemotherapy.
Future of bugs as drugs. How genetically engineered microbes produce chemicals beneficial to human health. With the rapid growth of medical science and technology, there has been an increased interest in developing a DNA memory device that will enable us to reprogram microbes' interactions. Microbiome engineering may be may be able to enable resident microbiota to supplement as functions of the kidneys and liver, since they take all the harmful chemicals in medicine and detoxify and metabolize the harmful chemicals. Ecological niche can be populated by engineering microbes, which then they release a specific molecule, antibiotics, anti-inflammatories, etc., to respond to a suitable stimuli, which will be infection, inflammation, tissue damage, etc. Scientists are currently testing microbes to cure a wide range of medical problems. For example, bacteria and yeast could one day be used to bypass more expensive options to treat malaria. Lactobacillus, a bacteria found in the gastrointestinal tract, may be genetically enhanced to manufacture proteins that target and attach to HIV. Special strands of the adenovirus, also known as the common cold, have been altered to deploy anti-cancer genes within tumors, killing cancer cells while leaving healthy ones unscattered. Scientists have created a vaccine that targets tumors that have already formed. Genetically engineered yeast in a vaccine delivered a common tumor protein that has stimulated the immune system of mice, thereby destroying tumors. The result of these animal trials may eventually help patients with colon, rectum, stomach, breast, or lung cancer. In the future, we may see bacteria Salmonella be genetically modified and used for killing cancer cells and attacking tumors. The particular strand of Salmonella enterica attacked tumors and destroyed cancer cells, the only problem scientists are facing in harnessing Salmonella's cancer-fighting abilities without causing infections in humans.